Welcome back. And I don't know about you, but I really need that moment of chill. Kind of want to just let that keep playing. Maybe I'll come back to that. Today, moving on, talking about biomolecules. These are molecules of biological importance. I think I need to fix the screen just a sec. Okay, so discussing biomolecules. Just like the name sounds, we can very easily break this down. Apparently not. Apparently my iPad is not. Oh, there we go. Cooperating with me. I don't know what's going on today. Okay, apparently I'm gonna just not be able to change colors. Bio refers to biological. Of biological importance, molecules are two or more atoms bonded together. There are 4 classes. You're familiar with these. We're going to go through them. When we're talking about these 4 classes, carbohydrates, lipids, nucleotides, and proteins, these are specifically organic biomolecules. So there are inorganic biomolecules. They're of biological importance, like, oxygen. It's pretty important to life. But carbohydrates, lipids, nucleotides, and proteins are specifically organic biomolecules. So that means that they contain carbon. I always feel like, and I think I'm just being pedantic and tacky, but when you see things that are advertised like organic food, like buy the organic carrots, and I always want to say, well, are there inorganic carrots? I know, but all this means is that they contain carbon atoms. So any of these organic biomolecules, you expect to see carbon. They're synthesized by living organisms, and these organic biomolecules, you can think of them as the currency of energy and growth. So if we don't have enough of any of these biomolecules, an organism can't live and can't grow. We're going to stop for just a second here, and I would like you to think about, what does the word carbohydrate make you think of? You may have thought of like bread or pasta, which is wonderful. These are foods that provide us with quite a lot of energy, and carbohydrates are energy sources. If we break down the word, carbohydrate actually means carbon and water, which makes it very easy to remember what it's made of. It's made of carbon and water. Carbohydrate is actually a 1 to 2 to 1 ratio of carbon and water. The simplest carbohydrate is a monosaccharide, and you've probably heard of these before: monosaccharides, disaccharides. So mono means 1, saccharide means sugar, so the simplest carbohydrate is 1 sugar. The key to it being a carbohydrate is that it can't be further broken down into another sugar, into a simpler form that is still a carbohydrate. A sugar is a carbohydrate. So this is what a monosaccharide looks like. Carbon and water ratio, C6H12O6. You might be familiar with that. And when we're looking at different chemical bonding as we move forward in the course, remember that we're talking about things that contain carbon, and carbon is kind of the key to our organic biomolecule definition, one of the keys. But very often when you see this structure, I don't know why my pen isn't changing colors. When you see the structure, the carbons that are here, that are shown here, are very often going to be absent. So what you'll just see is something like that. But just know that at each of these corners, there's a carbon. So there's the structure of a monosaccharide. If you add 2 monosaccharides, so you take 2 1s, you get a 2, right? all this is 1 plus 1 equals 2. Di means 2. Mono, mono, 2. Disaccharide. Sucrose is an example of a disaccharide. If you add a whole bunch of monosaccharides together, you get what's called a polysaccharide. And poly means many. You'll see that in a lot of places throughout science, through biological science. You'll see poly meaning many. So this is many saccharides. So lots of saccharides bonded together is a polysaccharide, and starch, one of my favorite things in the world, is a polysaccharide. All right, lipids. Let's stop and think for a second. What do you think of when you hear the word lipids? Many of us probably think of fat, right? Cool. When you hear the word lipid, you think of fat. Fats are actually made of something called a triglyceride. And you may notice there's 3, 1, 2, 3 triglycerides. And then here is the glycerol backbone. That's where it gets its name. Fats are made of triglycerides. And I don't know why that square is there randomly. I'm not sure what that's about. But you have 3 fatty acids. That's where it gets its name, the 3 fatty acids right here, and the glycerol backbone. So when you see this structure, you know that that is a fat. Fats are not the only lipid. Oh, there we go. What is that? I don't know why it's doing that. Okay, so fats are not the only lipids. The only thing that's similar between all of the different lipids is that they are very hydrophobic. And this is something that you're familiar with. If you pour oil in water, the oil is going to beat up because it's hydrophobic. So an oil is a fat, and this is true of all lipids, is that they are all hydrophobic. Other important lipids, eicosanoids. And eicosanoids, and we're going to talk about these more when we get to cell signaling, are absolutely critically important as cell signaling molecules. One of the most important ones is arachidonic acid. They're also really important in inflammation, allergy, fever, childbirth, and all of this is related to cell signaling. So they're carrying messages between cells, trying to trigger things happening in cells, and we're going to talk about that a little bit more in the next lecture. Prostaglandins might be one that you're very familiar with. So I think the most familiar eicosanoid is prostaglandins. Other lipids, steroids. This is my favorite one because I feel like a lot of people don't know that steroids are lipids. Steroids, every steroid, all of them, are synthesized from cholesterol. Synthesized from cholesterol. Every steroid is synthesized from cholesterol. And we tend to think of cholesterol as a really bad thing. But you have to have cholesterol in your body in order to synthesize the steroids. That as we talk about the rest of the body, particularly the endocrine system, you'll see how critical steroids are to the functioning of your body. You'll notice all of these different steroids. So hydrocortisone here, estradiol here, prednisone here. These are all different steroids. But they all have this one key feature. You'll notice that this 4-ring structure is present in every single one of these steroids. And so that is how you can always identify a steroid, is it has this 4-ring structure that it gets from cholesterol, because they are all derived from cholesterol. Then we have phospholipids. Phospholipids. I hope what just popped in your head is phospholipid bilayer, because we study that in many, many classes. The phospholipids, so here we're going to start integrating different terms that we've already learned. Phospholipids, the head of a phospholipid is hydrophilic. Hydrophilic means what? The tail of a phospholipid is hydrophobic. What does that mean? So we have a molecule that is water-loving on one end and water-fearing on the other end. So this part of the molecule likes water. I love water. And this part is scared of water, doesn't like water. What is that even about, having a molecule that, like, how is it going to work in the body? But think about that for a minute. How does that work? How do you have a part that's hydrophilic and a part that's hydrophobic and it's still functional in the body? And we're going to come back to that. Oh, So we already talked about hydrophobic and hydrophilic in the previous lecture, but now we're gonna recall that information about hydrophobic and hydrophilic. Other lipids, phospholipids. We already talked about that. Okay. So I said that we would talk about how this whole hydrophobic hydrophilic thing works. If you have a part of a molecule that loves water and a part of a molecule that hates water, what you can do is you can orient those molecules so that the part that doesn't like water is the interior of the structure and the part that does love water is the exterior of the structure. And that's exactly what happens with your cells, is your cells are a layer of two layers of phospholipids, where the part that loves water is on the outside and the part that not so much likes water is on the inside. And we're going to talk about that more in the next lecture. Nucleic acids. Okay, so nucleic acids get their name because they're composed of nucleotides. A nucleotide is a 5-carbon sugar plus a phosphate group plus a nitrogen base. Here is your sugar. Here is your base. Here is your phosphate group. I don't know if that looks familiar to you, but after you've taken genetics, it most definitely will. Nucleotides are the thing. They are the basis for DNA and RNA. If you look at the double helix, what you've got right here, so this phosphate sugar base structure here is happening here. So you have your sugar here. This is the sugar. And then here is your base, this guy right here. Here's your base. And then you can't really see it. Well, yeah, you can see it. There's your phosphate group. And this pattern, you can see if you look closely, repeats over and over and over and over again. And this structure, did I do that? No. So this part, I promise I'm trying to stop saying so. This part on either side is what we call the backbone of the DNA. It's the DNA backbone. And then the bases come across, and they're often kind of referred to like the rungs of a ladder. And so what I would like you to think about is when we're talking about a nucleotide, this structure, the nucleotide itself, which parts of the nucleotide make up the backbone and which parts make up the rungs? All right, so hopefully what you came up with was that the sugar plus the phosphate make up the backbone, and that the bases make up the rungs. So nucleic acids, they're arranged into this sugar phosphate backbone, and with the bases extending out. And in DNA, those bases bond to each other across the center of the double helix, whereas RNA, the bases just stick out. Bases here, see, they're not bonded to anything. Without bonding, it's single-stranded. Nucleotides have other uses as well, they're not just DNA and RNA. They also make up an absolutely critical molecule, ATP, adenine triphosphate. Adenine triphosphate is cellular energy. And we're going to talk about adenine triphosphate quite a lot throughout the course of the semester, particularly when we're talking about muscle and what we call the power stroke in muscle contraction. ATP and ATP being broken apart is critical to that, all of that happening. Moving on, proteins. What are proteins and where do they come from? When we talk about the cell, we're going to talk about this a little bit more. Or we're going to talk about where this happens, I should say. But the central dogma of genetics. Hope you can read my scroll there. The central dogma of genetics is that DNA is transcribed into RNA and translated into protein. This is the core of genetics. You get DNA transcribed into RNA and translated into protein. So any protein that you have in your body doing biological work was transcribed directly from your DNA and then translated. Your DNA is in the nucleus. This transcription happens there in the nucleus, and then translation happens in the cytosol. We're going to go through that again briefly, a cell review. We eat protein, but our body has to break it down in order for it to be useful. And then any proteins in our body that are actually doing biological work, our body uses components of the proteins that we ate. Of course, protein's critical to our health, but it has to make those proteins in order for them to be able to do biological work. What actually is a protein? A protein is made of amino acids. Really? Hang on. I'm going to see if I can get my colors working. Okay, fixed it, I think. What are proteins? So if you look here, what you see is this chain of little balls. And what those little balls are representing are amino acids. Amino acids are very frequently represented that way. Proteins are a chain of amino acids. Amino acids, a whole bunch. And which amino acid goes in which order in the chain determines what the protein is going to be. Protein is a chain of amino acids. What is an amino acid? An amino acid is carbon, hydrogen, oxygen, and nitrogen, and this is the actual chemical structure. What matters about this, or what matters, what makes an amino acid different is this group right here, the R group. All of this stuff, no matter which amino acid it is going to stay the same. And what makes an amino acid different is called the R group. When you make a protein, you take it and you put it together. You put a whole bunch of different amino acids together in a chain like this. But functional proteins aren't a chain. They look something like that. At least that's how they're modeled. So we've got all this what happening. I don't see any little balls in there anywhere. Well, proteins are wildly, incredibly complex. There are multiple structures to a protein. This structure where we have a bunch of little balls, just a line of amino acids, is called the primary structure. Individual amino acid sequence is the primary structure. Then the body takes that amino acid sequence and it folds it up. Folds it up into a primary structure or into a secondary structure. And that secondary structure can take two forms. It can be a beta sheet, that's this one, or it can be an alpha helix, that's this one. And a single protein can have some parts that are folded into a beta sheet and some parts that are twisted into an alpha helix. Then the body goes from that secondary structure. So primary structure was a line of amino acids. Secondary structure were folded into alpha and beta sheet, alpha helix and beta sheets, and then it further folds those alpha helices and beta sheets into the tertiary structure. And the tertiary structure is what determines the function of the protein. So here you can see this. There's an alpha helix. There's another alpha helix. Then here's a beta sheet, and here's a beta sheet. And what the body does is it takes those already twisted up parts and it folds them onto each other and it makes it three-dimensional. And the critical thing is that this structure, this tertiary structure, is what actually determines the function, because it determines what parts of this amino acid sequence are available for interaction with other things. Finally, you have the quaternary structure. And a quaternary structure is 2 or more polypeptide chains. So the body took that primary structure, folded it up, took that secondary structure, folded it up, and it did that with 2 different polypeptide chains, and then it stuck them together. So in this example, this would be 1 polypeptide chain that got folded up, and the different colors are exactly what that's representing. This would be another. Each of these colors represents a different original polypeptide chain that went through that process of primary structure, secondary structure, tertiary structure, and then they got bonded together to form a quaternary structure. And one of the most well-known quaternary structures is hemoglobin. Cool. All right, so that's biomolecules, and we'll pick up next with an overview of this cell.